So welcome everyone again to the Green Era Hub webinar series. My name is Barry Crosby and I'm one of the communication officers for the Green Era Hub project. If you've been here with us before, you'll know that the Green Era Hub project officially started in September 2022 as a coordination and support action of the European Commission under the Horizon Europe programme. And it currently represents 15 EU ERANETs, co-funds and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs in October 2023, and we've had 5 very successful series so far. The first one was circularity in agriculture. The second series was related to biotechnology to advance sustainable solutions for bioeconomy. The third was sustainable food systems, and the fourth looked at precision agriculture technologies for a sustainable future. And the one that we've just finished, in fact, last month was plant health and protection. If you're interested in listening to the talks to any of the topics that I've just mentioned from these series, our recordings are now available to view on the Green Era Hub YouTube channel. Today, we're going to continue our new Breakfast Club webinar series, which focuses on the topic of animal health and welfare. And for the next couple of months, we're going to be hosting a series of expert-led discussions aimed at fostering a deeper understanding of sustainable strategies for improving animal health, preventing disease, managing pests, and strengthening the connection between animal, human, and environmental well-being. And just a few housekeeping announcements. Please do feel free to type your question into the Q&A and chat section, which I've enabled for everyone. I'm going to do my very best to address as many questions as possible within the time that we have this morning, which is 1 hour. If at the end of the session you have a question for either of our speakers, please do remember to address the speaker in the question. Now, today we have 2 panelists joining us, Professor Javier Bezos and Dr. Keelan O'Driscoll, and we'll open our session this morning with Javier Bezos, Professor in the Department of Animal Health and Veterinary Faculty at Complutense University in Madrid, Spain, where he's currently an Associate Professor and Deputy Director of the EU Reference Laboratory for Bovine Tuberculosis. With a PhD in veterinary medicine focused on tuberculosis diagnosis in goats, Professor Bezos's research spans animal health, immunology, and disease surveillance. He is the principal investigator of the international imdiTBap project and Spain's Goat TB-Free initiative, contributing to national and EU level TB eradication efforts. He has authored over 110 scientific papers and has contributed to more than 350 conference presentations. Professor Bezos is also an associate editor for Frontiers in Veterinary Science, BMC Veterinary Research and Vaccines, and regularly advises the European Commission, EFSA, and DEFRA on TB control and animal health policy. Today, Professor Bezos' talk is entitled Improving Diagnosis of Tuberculosis in Domestic Ruminants, the imdiTBap project, which is part of the EU-funded International Coordination of Research on Infectious Animal Diseases. Javier, thank you very much for accepting our invitation to our webinar series. We're absolutely delighted that you're able to join us this morning. I'm going to stop sharing my screen now so you can start sharing your presentation. Thank you very much.
Well, first of all, I would like to thank Barry to give me the opportunity to present briefly some of the results of this project. The acronym of imdiTBap means improving diagnosis of tuberculosis through the use of new antigens and test platform. This is an international project that started in April 2023 and is planned to be finished next year, more or less in March of 2026. And probably, as you know, you are familiarized with the tuberculosis infection, and you know that the eradication of tuberculosis is a significant challenge, not only to animals, but also to public health, because since this is an important zoonosis. Also, you know that domestic ruminants are the main reservoirs of this zoonotic tuberculosis, mainly in low and middle income countries. And obviously, it means like the disease has a significant impact, not only on health and welfare, but also since the economic point of view on productions. There are many reasons that difficult the eradication of tuberculosis in ruminants, talking about cattle, goats, and buffalo, mainly. But one of these main reasons is the limited performance of the current and the modern diagnostic tests. Our hypothesis before starting this project was that the eradication can be accelerated by improved diagnostic tests that overcomes the limitations mainly associated with the use of the current PPD tuberculins, that you know that is the official antigens that we use for ante-mortem diagnosis of the disease in ruminants. As you probably know, these PPD tuberculins contains antigens that are shared between the tuberculous and non-tuberculous mycobacteria, and also these reactives can differ in terms of compositions and also in terms of quality and biological potency between manufacturers. The consortium of this project is made up of 8 partners from 5 different countries, more or less 12 senior researchers and 5 PhD students are related directly with the activities that we are performing in the project. The main objectives of the project was to improve the ante-mortem TB diagnosis through the development of an evaluation of more specific antigens. In particular, we are evaluating 2, the DSTF and P22. I talk about them afterwards, and new test platforms in cattle, goats, and buffalo. And the idea is to perform not only experimental studies, because we have a lot of information about the performance of these antigens in experimental studies, but our idea is to perform large-scale study under field conditions and different epidemiological situations in the different countries. So the plan is to perform at the end of the project more than 25,000 analyses in the different animal species and using these antigens and test platforms. The idea is to try to define new cost-effective diagnostic strategies for TB eradication in order to improve the current eradication programs. In this slide, you have a summary. I'm not going to go in depth in this information, but a summary of all the analysis that is expected to carry out by the different countries involved in the project, in the different animal species. And as I mentioned before, we are using 3 different antigens mainly. The current PPDs, the official reactives that we are using for ante-mortem diagnosis, that are compared with the experimental DSTF, that is a fusion protein containing ISAC, CFP10, and RV3615C, that are antigens specific of the Mycobacterium tuberculosis complex mycobacteria, and also an experimental reactive that is called P22, that was developed here in Spain in collaboration of 2 institutions, our institution and also Instituto de Salud Carlos III, that is obtained by immunopurification from the commercial PPDs. And as you can see in the slide, this P22 has a reduced number of antigens that are shared between bovine and avian PPDs, so a higher specificity is attributed to this antigen in particular. And also we are using these antigens in different tests, the intradermal test and also the interferon gamma release assay that you know that are the official tests that we are using now in domestic ruminant. And also we are trying to develop and validate other experimental technologies or platforms for diagnosis of tuberculosis that are a multi-cytokine platform detection based on Luminex technology, and also P22 ELISA for detection of antibodies, rapid test for detection of antigens, both of them not only using sera, but also using other samples, other matrix, like, for example, milk. And in parallel, we are developing and validating even diagnostic tools that are not based on immunological response, as for example the spectroscopy based on nuclear magnetic resonance for metabolomic analysis. I would like to show you briefly some of the results that we have obtained in the first half of the project. I would like to highlight some of them because it's not possible in 15 minutes to summarize all the results that we have up to date. But regarding the intradermal Maltese, we have tested mainly cattle and goats. And this is, for example, a summary of the results comparing the different antigens in TB-infected hares. In the slide, in the graph, you see the reactivity in TB-infected hares using the different antigens and different interpretation criteria. And as you can see, using the official test, the single intradermal test using the PPD, more or less the reactivity obtained in these hares. Regarding the comparative test, also official test using the PPDs, you can see that the reactivity is lower than using the single intradermal test, something that is expected because this test is more specific and less sensitive in comparison to the single intradermal test. But the interesting point is using the experimental antigens in these infected settings. As you can see, using the DSTF, this experimental fusion protein, the reactivity is more or less similar to that obtained using the comparative test, excluding an extra severe interpretation that is recommended by the manufacturers, that you can see that the reactivity in that case is very similar, even using the severe interpretation, or similar to that using the severe interpretation of the single intradermal test using the PPDs. And regarding the P22, the reactivity in these infected settings was the highest, was higher than that obtained using even the official test with the PPDs. Regarding the interferon gamma release assay, we have used two different kits for analysis, the Bobigan and IDO screen, and also we have used different interpretation criteria, as you can see in the slide. And well, regarding the Bobigan, as you can see in the slide, the reactivity using the PPDs was the highest, significantly higher in comparison to the DSTF and the P22, as you can see. This is related with a higher sensitivity in principle using these PPD tuberculins. And regarding the IDO screen, the differences were lesser. You can see that the reactivity in this case was very similar, excluding that with some interpretation criteria, even using the DSTF, the reactivity was slightly higher than using the PPDs. The interesting point is in goats, because in goats we have performed not only analysis in infected settings, but also in TB-free herds. And the number of animals tested is significantly higher in comparison in cattle, because, for example, in Spain, we have a lot of infected herds in goats, and it's more difficult to find infected settings in the case of cattle. And you can see in the slide the results using the intradermal test, both in TB-infected herds and in TB-free herds. Again, using the single intradermal test and the comparative test, the official test using the PPD, you can see the reactivity. And well, the interesting point here is that you can see that in TB-free herds, that less or that lower sensitivity attributed to the comparative test is related obviously with this high specificity that was perfect in infected in TB-free herds, sorry. And with the DSTF, the synthetic protein, the results are also very interesting. You can see that more or less the reactivity in infected setting is slightly higher than that obtained using the comparative test, but lower than using the PPDs. And in TB-free herds, the specificity is quite high, very similar to the comparative test, but using that extra severe interpretation that is recommended by the manufacturer that entails a very high reactivity in infected settings, also the specificity decreased significantly in TB-free herds. So probably this is not, at least in goats, a recommended cutoff point in order to keep a good balance between sensitivity and specificity. And regarding the P22, The results also very interesting because the reactivity was very similar to that obtained using the single intradermal test and PPDs in infected hares. But in TB-free hares, you can see that the specificity was significantly higher or better in comparison to the PPD. So they demonstrate a very good balance between sensitivity and specificity in these species. Regarding the interferon gamma release assay, also the results up to date are very interesting. We have also used both type of commercial kits, Bovigan and iDireScreen. And in the case of Bovigan, we have used samples from TB-infected setting and also from TB-free hares. You can see that in TB-infected hares, the reactivity was slightly higher using P22, who could be related with a higher sensitivity using this antigen in comparison to the PPDs and the STF. But in TB-free hares, there were no significant differences. Obviously, there was a slightly higher reactivity using the P22, but the differences were not significant, showing a very good specificity using the three reactives. And regarding the iDireScreen, it was interesting that in general, the reactivity was higher than using the Bovigan. But in this case, there was no significant difference regardless the reactive use for blood stimulation. You can see that the reactivity was very similar in infected settings, and also the specificity was quite high, or almost perfect in TB-free hares using or regardless the antigen use. Finally, I would like also to highlight some of the techniques that are not common for diagnosis of tuberculosis, at least in animals, and I would like to highlight some of the results that we have obtained in the context of this project. Probably you know that metabolomic analysis based on spectroscopy of nuclear magnetic resonance has been used previously in human health for diagnosis and prognosis of different infections, including tuberculosis. And we have the traditional equipments that are the high field or high resolution nuclear magnetic resonance equipment. These equipments, for example, are very expensive. It costs around 1 million euros. And also the cost of analysis and that kind of things and maintenance every year is very high. So this is a technology that has not been used previously in animal health. But in the recent years, the different manufacturers have developed like the benchtop nuclear magnetic resonance equipments that are like desktop equipments. These equipments has a reduced cost in comparison to the high field nuclear magnetic resonance equipments, and also the cost of analysis and maintenance is significantly lower. So this is a technology that could be an option to apply in animal health. In this case, or related to this, we previously published a study using this technology for tuberculosis diagnosis in cattle. And in the context of the project, in parallel, we have also made some studies to try to define the potential of this technology for diagnosis of tuberculosis in goats. In this study that is pending to be published, we use the two types of equipments in order to compare the accuracy of these benchtop or low field nuclear magnetic resonance equipments. And we use three different experimental groups, animals infected with tuberculosis, with paratuberculosis, and also control animals. And in the different charts, you can see that each point represents an animal and is distributed spatially depending of the expression of more than 1,000 metabolites detected in plasma or serum. It means that the points that are more near are more similar regarding the expression of these metabolites, and the points that are more far, each of them, it means that are more different regarding the expression of these metabolites. And you can see that using both types of equipments, the power of the discrimination between tuberculosis, paratuberculosis, and control animals was very high, showing that it could be also an option in the future to try to go in depth in order to define is this a potential diagnostic tool, antemortem diagnostic tool for diagnosis of tuberculosis in domestic ruminant. Also, I have not included information in the slide, but we have identified specific metabolites that could be used as biomarker, not only for diagnosis with diagnostic potential, but also with a prognosis valuable or useful tool. So in conclusion, I would like to highlight that this experimental antigen that we have used in the context of the project seems to show a better balance between sensitivity and specificity in comparison to the PPDs, the official PPDs, mainly in the skin test. In the case of the interferon gamma release assay, the antigens show a more similar performance. Also, we have obtained differences between cattle, goats, and buffalo, and also between, as you can see, as I have mentioned before, between the kit used, the manufacturer. Also, regarding the metabolomic analysis that I have briefly showed you, it could be, or it showed to be a useful tool for diagnosis of tuberculosis, and not only tuberculosis, but also other mycobacterial infections, for example, paratuberculosis. And it's very it's a very useful tool also to identify some metabolites. We have identified more or less between 10 or 15 metabolites that could be also useful biomarkers, not only for diagnosis, but also for prognosis purposes. Finally, it's something necessary to remark that we have obtained differences between animal species, and we are also using ongoing studies, not only regarding the result that we have obtained and that I have showed you in this presentation regarding the intradermal test mainly and the gamma interferon release assay, but also regarding other diagnostic tools that we are developing in the context of this project, as for example the P22 ELISA, this multiple cytokine platform that are developing our colleagues from AFA, and also the rapid test. So, this is more or less the things that I would like to show you about the project. So, thank you very much for your attention, and I'm open to any questions if you have regarding this. Thank you very much.
Javier, thank you very much for such an interesting and insightful presentation. I just want to remind everyone that the chat box and Q&A section is open. So please do feel free to type a question for Professor Bezos at any point. Javier, just while we're waiting for one or two questions to come in, I was just wondering if you can tell us a little bit more about the background to the project, the inspiration for the project. And I'm thinking particularly in terms of the current limitations of TB diagnostic tools in domestic ruminants, and how that kind of gave you the inspiration or idea for developing some of the things that you've mentioned and discussed in the imdiTBap project, and which you hope to overcome.
Okay, very interesting question, Barry. Well, really, the inspiration is based on the problem that we have in Spain regarding the control of tuberculosis, because you know that, in general, the situation about tuberculosis in Europe is very good, but we have some problems in some countries, including Spain. And in Spain, we have characterized very well the difficulties that we have for eradication. And among that factors, obviously, diagnosis is one of the main limitations. It's not the only one, or it's not perhaps the most important, but obviously something that is very important, not only to get the OTF status in Spain, but also or other countries, but also for the future. Always to try to research on diagnosis on tuberculosis, I think is very important, not only in the animal, but also in the human side. And in this case, regarding, for example, the PPDs, as I mentioned, that is the official antigen using now for diagnosis, is like a very old antigen. It has more than 100 years. And we know that the limitation regarding the quality and that, as I mentioned, the problem that it shares antigens between non-tuberculous and tuberculous mycobacteria, and that it can produce this interference in the diagnosis due to cross reactions, is something that we have to solve or try to improve. Obviously, it's not only important in order to improve the diagnosis, but also because, for example, the PPDs are evaluated in animal models. And well, the objective is to try to reduce the use of animals for evaluation of these PPDs. And with these synthetic proteins, these synthetic compounds that can replace the PPDs, we also can reduce the using of experimental animals for to evaluate the biological potential of the PPDs. That is something also very important, because in terms of compositions are synthetic, and it's very well known the reactives that are including these synthetic proteins. So it's something that in parallel we are increasing, or we are trying to improve the diagnosis, but we are trying to reduce the using of experimental animals for evaluation of the PPD tuberculosis.
That's fascinating, Javier. And I was just wondering, do you have any plans to train veterinarians or technicians maybe to implement the new tools that you're kind of advancing in this project?
Okay. Well, you know that, for example, I think it was also an idea that we get from your colleagues in Ireland, because we started I think in 2012 or 13, we started to do like a training programs or a training courses for all the vets that are involved in our education programs. Obviously, these programs are mainly based on theory, but it has like a practical component. And regarding the part that is based on theoretical classes, we include information about the development of these diagnostic tools and the activity that we are doing in the context of this project and other national projects that we are developing now. So I think it's something very important. It's not only important to train the people who is involved mainly in the field diagnosing tuberculosis to improve the official test, but also to transmit information that we are trying to improve this diagnostic test for the future. And it's something that we are doing every year in these training programs that are organized by our Spanish Ministry of Agriculture.
Thank you, Javier. Just one last question. Do you have any plans to validate the diagnostic tools across different breeds and species of domestic ruminants, for example, cattle or sheep?
Yeah, well, we started to do most of the analysis in cattle and goats, because most of the problem of tuberculosis, or the highest prevalence in the different countries, is mainly in this species. In the case of sheep, the prevalence of tuberculosis is very low, including Spain. But obviously, well, you know, we can say that the things that you can apply in goats could be applied in the same way for example, in sheep. But we are trying in this final year of the project. We are more focused on the analysis of all the information provided for all the countries that are involved in the project in the different animal species, to try to extract definitive conclusions about the performance of these antigens. Well, the objective is to transmit this information to the official veterinary services in order to say, well, this is something useful or not, depending probably, obviously, of the epidemic situation in each country or in each region. But if it is okay, we will try to do like a official validation in order to use these antigens officially in the context of this irrigation program. That is the final idea, obviously.
Javier, thank you so much. I think we unfortunately have to leave it. I could go on and go on for another half an hour, I think, with so many questions. It's a really, I think, a fantastic presentation and a brilliant study. And once again, thank you so much for taking the time out of your morning to join us and talk about your project. So now joining us is Dr. Keelan O'Driscoll, Senior Research Officer in the Animal and Grassland Research and Innovation Centre at Teagasc here in Ireland, an expert in animal welfare science, focusing on how husbandry and management practices influence the well-being and productivity of farm animals. Dr. O'Driscoll's work explores the feasibility of innovative production systems using animal-based and environmental measures, with an emphasis on reducing stress and improving welfare outcomes. She leads and contributes to several national and EU-funded projects, including OneWelPig, Fit to Farrow Free, and Welfare Partner, which explore high welfare pig systems, free farrowing management, and health welfare integration. Her research adopts a multidisciplinary approach to welfare, sustainability, and production efficiency, playing a central role in shaping evidence-based, welfare-focused practices in modern livestock farming. Today, Dr. O'Driscoll's talk is entitled Using a OneWelfare Framework to Develop Alternative High Welfare Pig Production Systems, the OneWelPig project. Keelan, thank you very much for accepting our invitation to our webinar series. We're absolutely delighted to have you join us this morning. Again, thank you so much for taking the time out of your day to do so. Without further ado, I'm just going to open the floor to you. I'm going to stop sharing my screen so you can start sharing your presentation. Thank you very much.
Okay, thanks again for inviting me. Delighted to be here today. And just to point out as well that I'm really presenting this on behalf of a much wider team. My co-authors are there, and most of them actually contributed more to the physical work of this than myself. So I hope I represent them well. So this is a project that started in 2022, funded by the Irish Department of Agriculture. And it's an all-Ireland project, so there's people involved in Northern Ireland as well. And the aim of it, what we're trying to do is use a OneWelfare framework to develop alternative high welfare pig production systems for the island of Ireland. So just to introduce you to the concept of OneWelfare, if you haven't heard of it before, it's very similar and extends the concept of One Health. So this means that it recognises the interconnectedness of humans, animals, and the environment they share, but specifically with regard to the welfare of these three components rather than just health. So overall, then, this is a quote taken from the paper that I've got there below. It provides a useful framework to ensure the development of fair and resilient livestock production systems. So thinking about this then in the context of pig farming, commercial pig farming in Ireland is very similar actually to many other countries in the EU. You can see a picture there of some pigs from our own pig unit in Moorpark. And when it comes to the welfare of the pigs themselves, pigs in commercial farms generally have a much more barren environment than the kind of scrubby wooded areas that they would have evolved in, or that feral pigs would live in, with very limited foraging opportunities. You can see there that pen is typical of a commercial system, and there's only a little rubber toy there for them to interact with, whereas a feral pig or a wild pig or a pig outdoors would spend about 60% of its time exploring and foraging. They also have a low space allowance relative to their home ranges, can be between 100 and 1,000 hectares. You can see there, again, pigs taken from the Moorpark unit in a finisher pen, and these pigs aren't even stocked at the legal they're stocked more loosely than the legal requirement. And then their interactions with humans are fairly few. They generally only interact with them when there's a need, when it comes to some sort of veterinary or economic reason, like moving them from stage to stage. And their interactions can often be negative, because particularly when they're very young, they experience things like teeth clipping and tail docking, and removal from their mother for weaning and such. So it's very difficult to meet pigs' behavioural needs in this type of system and to ensure good welfare. So this is not just ensuring that there isn't poor welfare, but to actually give animals or the pigs themselves a good quality of life, or a life worth living. So other challenges then in the pig sector, and for farmers in general, really, not just pigs, is there's ongoing pressure to maintain or increase output, but become more sustainable, particularly when it comes to the environment. And this is reflected in policy, which is increasingly directed towards encouraging organic systems at EU level, agroecology systems, etc., which is very, very different, particularly in Ireland, from the way we currently farm pigs. There's also concerns about farmer well-being. This is just a recent paper. In Ireland, dairy farming is by far the most profitable agricultural enterprise, and yet 75% of Irish dairy farmers experienced work-related stress, and this was higher with a higher level of training, which was associated with more production output, so the more productive farms. There's also increasing public concern about animal welfare. This is repeatedly demonstrated in Eurobarometer surveys, and this is just another quote from an Irish study where consumers were concerned that they never really saw pigs, which led to questions and suspicion about the systems. And even though participants knew less about pigs than other animals, other farm animals, they did express concern about living conditions and housing and describing cramped indoor conditions. So all of these challenges, if they're not addressed, might damage the social license to farm animals. So the vision then of OneWelPig was to identify production systems that work with pig biology and are a viable higher welfare alternative to intensive commercial systems. So as well as improving pig welfare, we hope that this could offer farmers a viable means of income diversification with potential to capitalise on premium prices for higher welfare pig meat and contribute to the regeneration of the local physical and social environment. And this is just a schematic of the larger project, but today I'm going to talk to you about task 2, which was characterising the outdoor pig production sector in Ireland. So our first step was to make contact with people working in this sector. Up to that point, Teagasc did not have any contacts in the sector, and neither did our partners in UCD, Queen's Belfast, or in AFBI in Northern Ireland. So we developed a survey that we distributed online. We used a lot of social media like Twitter, Instagram, Facebook. And then Ophélie Manon was the postdoc that worked on the project. She scoured adverts for people selling pigs, looked up Instagram posts for people selling meat at farmers markets, etc., and contacted those people directly as well. So the survey was made up of, most of it was multiple choice, kind of very factual questions about the farm and the farmer, the husbandry and health status of the animals. But we also had a few qualitative questions, just where farmers were asked to describe their farm, use free words to describe their farm, what the pigs enjoyed and didn't enjoy, and their challenges and changes that are needed in the sector. So after we sent it out, in the end, we only got 90 usable responses, which is a large enough number, but still we know from the Department of Agriculture that there could be up to maybe 700 or 800 small farms in Ireland. You can see the distribution there on the map of where the farms were located. So most of them down in the south, but we got pretty good coverage of the island. From the answers, we were able to determine that most of these outdoor pigs were kept on pasture and in agroforestry systems. You can see there a farm that we visited. They could really be categorized as backyard rather than large commercial, because the average number of pigs was 35, usually with only 1 or 2 people working on them, and over about 40 hectares, with a large amount of variation there actually. So we also asked farmers why they actually raise pigs outdoors. And I just want you to note there that the dark navy color is, they were asked to rank things from not important at all to extremely important, which is the navy. And these were the options that they have. And you can see here just on those little graphs that when it comes to the reasons that were considered extremely important most of the time were related to animal welfare. So the farmers considered that it offered better welfare for their animals and it helped the animals to have access to the 5 freedoms. So that was the reasons why they keep them outdoors. But then the actual purpose for keeping the pigs, what was that? And it was almost a 50. Now, these were not mutually exclusive. That's why the percentages don't add up to 100, because people could keep pigs for more than one reason. Overall, in the survey, it was about 50-50 between personal meat consumption or rescues or land management or some form of commercial reason for selling pigs. So there, about half of the people ticked these boxes. And these are the people that we were particularly interested in for the purpose of this study. So we selected a number of farms and visited 20 of them in the winter and 20 of them in the summer to get more detailed animal and environment and human-based measures ourselves. So this is Nipuna, the master's student that worked on the project. So the animal welfare measures then we took were very typical of the type of measures we would take in indoor systems as well. So we looked at body condition score, the lameness of the animals, presence of parasites, skin lesions, including tail and ear lesions, which is where commercial pigs get a lot of injuries, and tear staining scores. With regard to the environment, then we only had 2 measures here. We used the shear vein test to look at soil compaction, and then we also looked at the diversity of plant species on the farm. So for the plant species diversity, we selected, it's in navy here and down here in the paddock, a no pig area where there were no pigs as a control comparison on each farm. And then we looked at the plant species diversity, both the foraging area that the pigs used and the defecation area within their paddock. And then regarding the humans, we used the World Health Organization quality of life questionnaires. We had a social scientist on board that furnished us with these surveys, and the data went directly back to her rather than the farmer having to answer the questions to us. So these questionnaires is made up of 20 to 25 questions, I think, and these were summarized then into 4 main categories: physical and psychological health, social relationships, and the environment. And then there was an overall score of 0 to 5 given as well at the end. So first of all, just a summary of some of the animal welfare data. So the data in the blue table there is what we got from the survey. So when it came to, we asked the farmers what health conditions have they observed over the last 12 months, and very positively, nearly 70% of them said they hadn't really observed any obvious health conditions. When we went out onto the farms, we observed that we never saw teeth docking, teeth clipping, tail docking, any tail lesions or ear lesions or ear necrosis, which is very positive, especially relative to indoor systems. So that ties in with what the scientists saw. There's a good correlation there between what the farmers and the scientists perceived. Lameness then was the disorder that the farmers saw most often, and we had a 0 to 5 scoring system, with 0 being no lameness and 5 means the animal couldn't move. And very positively, again, the vast majority of the animals that we saw when we went out scored 0, so no lameness at all. This shows that the farmers were actually picking up a bit more lameness than we were, but this could be because lameness can sometimes be very brief in duration, so just 2 visits over the course of a year was probably not enough to pick that up. But it does mean that, again, the farmers' perception was more or less in line with the scientists, or at least they weren't underestimating things. Then the next 2 things were to do with nutrition, so pigs being either overweight or in poor body condition. So this is the body condition scoring scale we used. And you can see again, when we went out onto the farms, we saw the vast majority of pigs were good or very good, with a lower proportion there being moderate, but none very fat, and very, very few animals in poor condition. So again, this means that the farmer's perception was more or less similar to the scientists. And these results are quite positive, because there can be situations where sometimes farmers don't recognize or perceive welfare issues on farms because they become accustomed to seeing them. So there's an expected level of welfare deficiency, whereas scientists tend to pick it up more. So it was quite positive that in this scenario, the farmers seemed quite tuned in with their animals. Now, when we asked them what the pigs like and dislike, for liking, the words that came out most often was freedom, and this was related to roaming. It's the social group, the freedom to pick their partners, exploring, foraging, and wallowing, which are all very natural behaviors for pigs. And when it came to what they dislike, then weather came out the most often, and this is related to wet and rainy weather, cold weather, and whether that's too hot. So this leading to thermal discomfort for the animals. So in summary then, I didn't list all of the measures we took, but the pros that we found regarding outdoor pig production in this type of system in Ireland was that the pigs could express natural behaviour. They were less likely to develop harmful behaviours like tail and ear biting than commercial pigs, low levels of aggression or skin lesions, there were lower input costs of the farm, and they were often integrated with crop rotation or forestry, so they were providing ecosystem services. The cons then, we've actually already published a paper that was looking at parasites, and we found that there were relatively high levels of endoparasites in the pigs. There were some lameness issues. They had experienced hot and cold stress more than indoor pigs. There were some evidence of feeding management was difficult for the farmers, some environmental issues associated with poor weather, and then lack of support in general for the industry when it came to advice and financial support. So moving on from that, then we had kind of our basic data in each section, and we wanted to try and figure out a way that we could kind of integrate everything together and see if there are any patterns across the farms when it came to humans, animals, and the environment. So we decided just to use a single visit per farm for this. We selected the summer visits. So this was between July and October 2023. And this gave us data from 223 individual pigs inspected across 29 paddocks. The number of paddocks varied across farms. And we only used, we didn't use pre-weaned pigs or anything. We used pigs from 3 months old to 6.5 years old. There were lots of different breeds. You can see here examples of some of them. So we didn't really use breed in the analysis because there were just too many for us to get any solid data. Now, this was work that was carried out by Ophelie, the postdoc in the project. She decided to use principal component analysis to try and pick up patterns or clusters of farms. So this is what she found. Now, these are preliminary data. It has been presented at a conference, and we're going to do a bit more kind of in-depth work on it. So I'm just showing you a very first view of this. But basically, we found 3 clusters. Cluster 2 with 5 paddocks in it, cluster 4 with 1, and then cluster 20 with cluster 3 with the remaining 20, cluster 1 with 4, and cluster 3 with the remaining 20 paddocks. And what we did then is we looked at the different metrics across the different clusters. So first of all, looking at animal welfare, we found that cluster 1 was the one that had the most pigs, or the highest lameness score on average. There were scores of 0 in cluster 2 and 3. Similarly, cluster 1 contained animals with the highest skin lesions, with the highest ear lesion scores, and also with the highest tear stain scores. So overall, cluster 1 had animals with the poorest levels of animal welfare based on our measures. Although I do have to say as well that even though these were worse than cluster 2 and cluster 3, the scores were still could still be considered low overall. So not bad welfare in general. Then when it came to the environment, first of all, looking at plant diversity, and just to explain these graphs a little bit, you can see there's a 0 line across the middle. So what we're looking at here is the difference between the no pig area and the foraging area on the left-hand side and the defecation area on the right-hand side. So anything below the line means that the foraging area was less diverse than the no pig area, and anything above means it was more diverse, and similar for the defecation area. So you can see here, cluster 2 was the cluster that had, when it came to plants diversity, it was much less diverse than the other clusters, with cluster 1 being very similar to the no pig area. And then when it came to the defecation area, cluster 1 actually had more diversity than the no pig area, whereas the other 2 clusters were below the line, so with less diversity. And then with soil compaction, cluster 1 again, the soil compaction was quite similar in the foraging area and the no pig area, whereas in cluster 2, the soil was much less compact in the foraging area than the no pig area, with cluster 3 intermediate, and a very similar pattern in the defecation area, other than cluster 1 actually was more compact than the no pig area. And this somewhat makes sense because you could see this pig here. If a pig is doing a lot of rooting, that means there's going to be very little vegetation in the area, and also the soil is unlikely to be compacted because the pig is rooting it up constantly with its nose. So then looking at the quality of life, we have our 4 areas here. Now the differences were significant for all areas, where cluster 2 scored the lowest for quality of life across physical health, social relationships, environmental health, and psychological health, and also overall. So how to define these clusters then? Cluster 1 we considered to be the one, kind of the pro-farmer cluster. So it had the lowest levels of animal welfare, even though they were still good, they were lower than the other 2 clusters. There was least damage to the environment, but it scored best for farmer quality of life. Now it has to be noted, there were only 4 out of 29 paddocks in this cluster, and they were all sow and boar paddocks. Cluster 2 then, where there were 5, this is kind of the cluster that was associated with the best animal welfare. You could see a pig there lying down in the grass in the sun, but it also was associated with most damage to the environment and the poorest farmer quality of life. We did note as well that all 5 of these paddocks were either organic or non-GMO feed farms, and it can be quite stressful to manage a farm that's organic or non-GMO feed because it can be difficult to obtain the feed, and then it's very expensive. So we thought that this could be contributing to perhaps some stress levels for the farmers. And also, if the feed was difficult to obtain, the pigs might have been carrying out more rooting in the environment and searching and foraging for food. Cluster 3 then was the remainder and was kind of average for everything, but still, particularly with animal welfare, still at good levels. So summarising this then, overall we found that the welfare of pigs was good. The farmers that we spoke to or that answered the survey were motivated by pig welfare. This is why they were carrying out their farming in these extensive systems. And also what was really nice to see was that the farmers' perception of pig welfare was reflected in the scientific evaluations that we carried out on farm. So it was somewhat unsurprising that we saw the impact of pigs on the diversity of plant species. Again, you can see a picture of there. Pigs will spend so much of their time rooting and foraging that is, unless they've got a very large area, the soil will be disrupted. Lots of rooting then will mean the soil is less compact, with fewer plants in the area. Now, we did have a very low number of paddocks and farms, really, for this type of analysis. There was a lot of diversity between them, and this would have impacted the results. And hopefully in the future, we'd be able to gather more data across more farms, a bit more balanced as well, to try and improve these. But the study has provided a lot of novel information. This was the first time that a study had been carried out on the island, going out to meet these farmers, and this will be used to frame future questions and project proposals. So I guess overall then, to sum it up, we think that the One Welfare Framework could be adopted to investigate the interrelationship between aspects of sustainability in pig systems to ultimately progress towards fairer and more resilient outdoor pig production systems and optimize welfare for all. And I would just like to acknowledge our funders and say thank you, or as well acknowledge the farmers that answered our survey, led us onto the farms and their pigs. And we had many placement students as well who helped us during the study. So thank you to those too.
Aileen, thank you so much. Again, another really interesting and insightful presentation, really clearly explained. Thank you so much. I just want to remind everyone that the chat box and Q&A section is open. So if you have a question for Dr. O'Driscoll, please do feel free to type a question in the chat box or the Q&A section at any stage. Gillian, I'm just going to ask you just a little bit of this, a similar question to Javier with regard to the inspiration for the project. And I'm going to ask you that question, particularly in relation to the kind of main welfare issues that exist in relation to conventional pig production, and how that kind of inspired your project, and how, I suppose, it gave your project the kind of the focus to address those particular issues.
Well, I suppose pig production, certainly in Ireland more than any other sector, it probably would be one of the more intensive ones. There are a lot of challenges when you're dealing with systems that are designed to be within concrete walls. I guess there have been changes over the year. You have a concrete facility and then it's static for 25, 30 years, but you've changes to the pigs over the years. It's changed genetics. We've got increased litter sizes. So there's many more pigs now coming out of each sow every year than there would have been before. So this leads to very practical problems for farmers, things like overcrowding, things like managing slurry and emissions. And then consequently as well, there's an impact on the pigs because any kind of changes that involve kind of more intensity generally will lead to animals being a bit more stressed. And this manifests then, I think, probably the biggest welfare issue for pigs, or the biggest obvious kind of demonstration of stress and a welfare issue is tail biting, which is an area that I've worked in for about 10, 11 years now. Really, really difficult to solve because it's so multifactorial. It's really a farm by farm situation each time, you know, if you've got a problem with it, to go in and find out what's causing this kind of an issue. This tail biting as well has kind of been the focus of a lot of work in the EU, both kind of when it comes to policy and research. And I think especially, you know, with the start of the Green Deal and the end the cage age stuff, things that were driven by policy and consumers, we kind of thought in this country, maybe it's time to start looking at alternatives and seeing if we kind of help farmers, both conventional and alternative, to try and find ways that they could maybe reduce stress levels for the animals, and this could then have a positive impact for themselves and the environment. There is a component of the OneWelPig project that is focusing on typical commercial systems in Ireland, so looking at ways that we can provide them with more environmental enrichment for the pigs throughout their entire life cycle, and kind of still concrete walled facilities, but with more access to fresh air and solid flooring and comfort for lying and things like that. So we haven't forgotten about the conventional farmers and want to kind of help them as well. But the outdoor sector was something that had never been looked at really in Ireland. And it's small, and we just thought maybe it's about time, with kind of growing interest in this area with policy wise and with consumer, that we should start trying to engage and learn what they're doing and maybe come up with some strategies to help and support them as well. So that was it, really. Very long-winded.
Thank you so much for that. It was really fascinating. I was just wondering, just you're discussing there the idea of creating that kind of balance between the reality and the ideal. And I'm just wondering, how does the project kind of balance trade-offs between animal welfare, economic viability, and environmental sustainability?
That's a really hard question. We do have other tasks as well where we're carrying out. There's a large task being managed in Queen's on life cycle assessment. So that's carrying out life cycle assessment exercises with both conventional and alternative systems when it comes to economics, the environment, and social life cycle assessment as well. So we're gathering in data from farmers, they're answering questionnaires and providing interviews to feed into that kind of a model. And that's actually only ongoing at the moment. We'll finish sometime next spring. So we'll have a few more answers there as to how they'll all balance up against each other. We also carried out, there's another task that was led by UCD, which was a land modelling exercise. So looking at a few different scenarios, if we transitioned from most of our pigs being indoors in conventional systems to different proportions of the country being used for agroforestry systems for pigs, how then would that impact land use and the environment across the island? And again, that's still underway, but we will hopefully have results there in the autumn. So we're trying to cover a number of different aspects. The project won't finish until about this time next year, so we're still waiting for results. Yeah, it's difficult to balance them all up. I mean, particularly with pigs, I guess, when you have the conventional farms, which might be considered or are generally associated with more welfare problems, tend to be a bit more environmentally friendly because they're not using as much land, and you get much more efficient production out of them than you would with extensive systems. So trying to find a balance between them both is different, I guess, or difficult.
Sure. And Keelan, sorry, just one last question just before we finish up. What would you say have been the biggest challenges in convincing stakeholders, especially farmers, I suppose, to adopt these systems?
Well, we're not really trying to persuade anyone to do anything. Because I don't, the type of what I just described to you there, that's completely unfeasible for a typical pig farmer in Ireland. That would be more kind of, I think this small scale outdoor pig production might be something that maybe a dairy farmer that wanted to diversify could like use a small bit of their land. Or that, as I showed you, one of the pictures there was some pigs in a forest. This was a guy who his business was forestry, and he used pigs then to clear the area in between the trees, which they did a very good job of. So just to kind of demonstrate that these animals could be used out there, this is what we like the ecosystem services part of it, that they could be used to do a job, and then you kind of get the pigs themselves as a byproduct almost. For the conventional farmers, we have tried to look at different options, for the enrichment and more space. And that's still in progress as well. I guess with the enrichment for pigs, one of the things that we were trying to do is demonstrate that you can use loose material for them in a rack. Irish systems are fully slatted generally, so it's very difficult to provide rooting material for the pigs on the floor like they do in some other countries. And just to demonstrate that if you put it into a rack on the side of the pen, the pigs will still use it and engage with it, and it will lower the risk of tail biting. And also looking at space allowance as well. To show that when you reduce the space allowance in the pens, this is what we found anyway, that you actually get much more efficient pigs and faster growth rate. So it can actually, there's a trade-off because you might have fewer pigs, but then they're more efficient with their feed conversion and they're growing more quickly and getting to the factory faster. So that's a benefit. So I guess really just trying to demonstrate those benefits of providing kind of conditions that are a bit more welfare friendly for the pigs, with the hope then that people will adopt and take them up. So we have one of the tasks again is a large, it's a knowledge transfer task, but most of what they're doing in that task is there's a stakeholder group that are trying to develop a roadmap towards higher welfare systems for pigs in Ireland. And that includes farmers from both conventional and alternative systems. So they're all kind of working way together at the moment as well. To try and see if they can identify a way forward and bring people along with us, I guess.
Keelan, thank you so much for answering the questions and again for such a wonderful presentation. Unfortunately, we do have to stop it there. It's just coming up on 10 o'clock. I'd just like to thank once again our excellent panelists this morning, Professor Javier Bezos and Dr. Keelan O'Driscoll. for giving us, I think, wonderful and insightful presentations and providing us with tremendous overviews of the respective projects this morning. Our next webinar will be on June the 19th, and I'll be sending the invitation out to all of you in the coming days. In that breakfast club, we will have Professor Romain Vollmer from the École Nationale Vétérinaire de Toulouse in France, talking about the identification of factors driving the emergence and spread of avian influenza viruses with zoonotic potential. And we'll also have Professor Beatriz Prieto Simon from the Catalan Institute of Chemical Research in Spain, discussing channel-based biosensors to support a precision agriculture approach to improving bovine mastitis management. Again, thank you very much to our panelists for joining us this morning and for giving up their time so generously. And thank you everyone for attending and for your questions. We really hope you enjoyed this morning's webinar, and we hope to see you with us again next time. Thank you very much, and have a good morning, everybody.